We were talking about the sarcomere, which is the functional or the contractile unit of the muscle fiber, and I was talking about the Z line and referring to that as this is the area where you actually will have this overlap of thin and thick filaments. That's fairly true, but a truer point, I mean, it is true, but the contraction doesn't go all the way to the Z line. The Z line is actually what defines the sarcomere. So you have the I band, which is going to have your thin filaments, and you actually have I band to I band, but that Z line that's in the middle of the I band is actually going to define, I guess, bisect the I band, but define that particular sarcomere. So sarcomere is considered from Z line to Z line. And what happens is the contraction happens with the thin and thick filaments sliding past each other, and they will go almost all the way to the Z line. But if we move on to the next slide, you can see that you have some other players in there that will prevent it from going all the way to the Z line. But this M line right in the middle, there are never going to be any thin filaments. It's always going to be thick filaments. Thick filaments, or actually we'll talk about the thin filaments first, or back to the Z line. This is actually made by a protein called actinin, and it stabilizes the sarcomere and basically designs a sarcomere. So the reason I'm saying this and kind of belaboring this point, I do want you to get the idea that this is one unit in and of itself, one closed unit in and of itself that happens to be working with every other unit that is next to it and is making up the billions of units that are making up this particular muscle. The structure of the thin and thick filaments can go even more microscopic. What we have here is a artist rendition of a thin filament, and the thin filament is made up of filamentous actin, which is called F-actin, which is actually a twisted row of two globular G protein or G actin. And the thing you think about the G actin, this is where you have the active sites where the thick filament is going to be able to bond to the thin filament and actually pull it to make the contraction. So that doesn't happen all the time. So these active sites are, the structure of this particular filament is created by this nebulin, which works to hold this actin strand together. But these bonding sites are controlled, I guess, or maybe regulated by this tropomyosin, which prevents actin and myosin interaction, so prevents the interaction between thick and thin, and then basically a lid on the tropomyosin, which is tropotin. And tropotin is a protein that is going to bind to tropomyosin, which is actually on top of the active site, and keep this muscle from contracting. So when you have the tropomyosin tropotin complex attached to the active site, the G-actin site, no contraction is going to take place. But when that has been released from the active site by the presence of calcium, so a calcium ion, then what you have is the setup for the thick filament to actually attach to that active site and then pull this muscle and create a contraction. And now to talk about the thick filament. It actually is attached to the Z lines by this protein called titin. And what titin does is not only gives stabilization to this sarcomere, but also is a very, it's drawn very well in this artist rendition. It's a very springy protein, so it allows stretch and then back, moving back into its original shape. So this tighten will allow this muscle to contract, and so it will actually condense itself, I guess you might say, like a slinky, condense itself, and hopefully I'm not totally, I think slinkies are still around. And then as the muscle goes back into the relaxed state, go into the state that you see here. So you can have a state where it actually, these loops, these kind of teal loops get closer together and then relax. The arrangement of the thick filaments is such that you have a tail, you have a head, just like you see here, and then you have a little connection between the tail and the head, and that's called the hinge. Thick filaments are made up of multiple of these units twisted together, and in the core of the twisting together you have these titin proteins, and then the titin proteins are attached at either end via this Z line, and remember the Z line is made up of a protein. And the head is where you have the binding site to the thin filament via that G-actin binding site that then is going to be exposed in the presence of a calcium ion. And the image here is showing how that pivoting goes on, and if you have the head of the myosin attached to the thin filament and then it pivots, it causes the thin filament to move closer to the M line but not in the M line. So it moves closer to the M line but not in the M line. We call this attachment of the thick filament to the thin filament a cross bridge, and that cross bridge causes the movement causes pivoting and produces motion. This image again comes from your book. This shows a contracted muscle, and what you have is the thick filament is still the purple, thin filament is the reddish color, and you have the thin filament moving toward that M line. You have the H band in place, but what you have is a diminishment of the I band. The I band almost goes away, and that contracts the sarcomere, contracting the entire muscle, because this action takes place over and over and over again, and that repetition, remember all of this is happening simultaneously due to the electrical impulse that's being passed through the entire muscle via that sarcoplasmic reticulum, the triad, and a contraction takes place. This wasn't, strangely enough, this whole thing wasn't known forever. There were a couple of gentlemen who looked at this and figured out that this is how a muscle is contracting, and created a series of experiments, tried it, published a paper. That paper ended up winning the Nobel Prize. It is called the sliding filament theory. So this discovery and this understanding of how a muscle contracts because you have the thin and thick filaments sliding past each other in a very, very predictable, regular way, was published and understood and studied, and that's what the theory is, the sliding filament theory. It really revolutionized and even, I guess in a way, maybe gave birth to what we know now as physical therapy. Because prior to that, certainly people were working on the muscles, but this gave even more scientific knowledge to how muscles work. If we know how they work, we're much better at making them work better and repairing them if they don't work. Then on to a series of other types of concepts. Maybe back to, so that was how the muscle contracts, but back to how does this electrical impulse actually get to the sarcoplasma via the membrane that's around the cell, and how does that become how does the muscle know to do that? We said these are voluntary muscles. You can choose to move them, but you choose to move them because the nervous system is regulating it. And so what you have is a space between the muscle system and the nervous system, and that space between the muscle system is called the neuromuscular junction. So neuromuscular junction you have. And what you see here is that the yellow color, and this picture doesn't come from your textbook. We'll get started on this picture. It doesn't come from your textbook, but what's in your textbook is the same thing. They've just divided it up. I wanted it all in one slide. And so you have the nerve that is this motor nerve that is going to feed this particular muscle group, this group of fascicles, and along the nerve is an action potential. Action potential is the sum total of all of the transmembrane potentials of that particular system put together, that particular nerve. So nerves have action potentials, and so do muscles have action potentials, but action potential tends to be the sum total of the change in transmembrane polarity all the way along the nerve. So that action potential reaches the point where the nerve comes very much in close contact with the sarcolemma, but doesn't touch the sarcolemma. So you have nerve system, you have muscle system. Two different types of tissue, two different systems. They don't touch each other. There isn't any place where they have fused contact. But what needs to happen is there has to be something that can bridge the gap, can have the nervous system talk to the muscle system. And those are called neurotransmitters. And so what you have is the action potential making its way all the way to the axon terminal of the motor neuron. So this is the nerve that's going to innervate this group of fascicles, and it ends at an axon terminal. That action potential causes neurotransmitters that are already sitting there in vesicles to fuse with the membrane of the axon terminal. Fuse with the membrane of the we call that the synaptic cleft, the membrane of the synaptic cleft, and then drop those neurotransmitters into the space between the nerve and the muscle, and that neurotransmitter happens to be called acetylcholine. If it's a muscle contraction, it's acetylcholine. Acetylcholine then is going to attach to the membrane or the folds of the sarcolemma. So acetylcholine is going to attach to the folds of the sarcolemma. That then is going to cause sodium potassium to be pumped out and sodium to go into the sarcoplasma. And sodium rushing in is going to change the polarity of the sarcoplasm. So that, remember, the sarcoplasm is the liquidy type portion of the fascicle, and this is going to be aided by calcium. And remember, calcium, or maybe I didn't talk about this yet, calcium is just housed right outside outside of the sarcoplasma. And so it actually isn't stored in, it's stored basically